Hello, everyone. Good morning, good afternoon, good evening to you, wherever you are in this world. We want to welcome you to today's webinar, See What We Analyze and Sort at UCD Conway Institute. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, please visit beckman.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Now, without further delay, I do want to welcome our speaker, Associate Professor Dr. Alfonso Blanco, Director of Flow at UCD Conway Institute at the University College Dublin. Also joining us for our live Q&A today, we have Dr. James McCracken, Commercial Product Manager at Beckman Coulter Life Sciences. Dr. Blanco, welcome. You may now begin your presentation.
Thank you. All right. Hello, everyone. My name is James McCracken, and I'm the product manager for flow sorters at Beckman Coulter Life Sciences. And it's my pleasure to introduce this webinar presented by Associate Professor Dr. Alfonso Blanco. Alfonso is the director of Flow Cytometry Core Technologies at University College Dublin and the Conway Institute. I think we all know Alfonso as a tireless and generous educator in flow cytometry, and I'm very happy to have him here today to give us a view of what makes a sorter a fit in his lab and the breadth of the applications that are being performed at the Conway Institute. So with that short introduction, thank you again, Alfonso, and please take it away.
Thank you very much, James. It's a pleasure to be here, and thank you for the invitation to share our experience with the cell sorter and the analyzer, and share the experience that we have in the UCD core facility for flow on what we are doing, and explain a little bit what the different researchers that we have in the core are working and using this. And this opens possibilities for our users to expand the possibilities of communicating their staff, but also at the same time looking for collaboration. So thank you very much for this opportunity. And I will start with this idea of see what we are analyzing and sorting at UCD Conway Institute. First of all, I want to make a disclaimer. So basically, as you can see, whatever I say, you can blame me. Do not blame Beckman Coulter. It's my opinion, and you can see there what we are saying. So this is a long history. I started in May 2004, so it's almost 20 years. We are in fact celebrating the 20th anniversary of the Conway Institute, and we started with this small lab with two instruments, an analyzer, the Coulter here, and also the cell sorter, the fax area on the other side. There was more and more demand for usage, but also different applications. So you can see how we expanded in the portfolio of the different instruments and different platforms in the facility. So we ran out of space, and what we need is more space for the users as well. So we move into this big, large lab. So again, things are changing, methods are changing, instrumentation is changing, and nowadays we have changed. Even the location is the same. We have to readapt to the new needs. At the moment, we are covering 38 different research groups in 7 UCD schools, but also we cover a lot of different universities here in Ireland and companies. So what we have at the moment in the facility are 4 different analyzers, 2 CytoFLEX S and 2 CytoFLEX LX. The CytoFLEX LX, one is the ultraviolet laser configuration. The other one has the near UV configuration. Then we have 2 sorters, the fax area 3 upgraded, and the CytoFLEX SRT. This one is placed into a biosafety 2 cabinet. We also got an IncuSite that we have inside an incubator just to look at the cells grow. Also, we have the Seahorse to look at the kinetics of our cells and look at the respiration. Also, we have the Luminex MagPeaks, and finally, we have the NanoSite NS for nanoparticle tracking. So as you can see, we have a good range of different instruments, and this is basically to cover the needs of our users. But you can see there that there's only one person, and that's because I have to run all of these. So what we do, as James pointed out, is education, education, education. I try to make sure that all the users, they know how to use the instruments from the beginning to the end. They can turn on, they can turn off, they have to clean and operate and even troubleshoot the instruments. So I'm looking always for instruments that they're very easy to use, but at the same time quite flexible and very, very powerful. So this is why we go to conference like the ones organized by ISAC, the International Society for the Advancement of Cytometry, CYTO. And in 2013, I find out a new instrument, a new device that called CytoGene, created by a Chinese company, a brand new company. And that was quite revolutionary from my point of view because of the capabilities that this instrument had. And then, which ones are these capabilities? First thing, the detection system. I thought that was extremely sensitive. As you can see on the portfolio, they were saying that they have 78 times more sensitivity in some detectors like PERCP Psi 5.5. Filter configuration was quite a good range of different filters. You can see filters are quite small. And what I was shocked was the space that this instrument was occupying. It was based on an Acri C6 that I have previous experience with this one. So I was quite understandable why we have this also large dynamic range into the detectors. So I easily talked to the person in charge, John Chen. I asked for a beta testing, but things changed slightly because in 2014, this company had been acquired by Beckman Coulter, and this is the platform of the CytoFLEX. So this was presented in 2014. Later on, we got the instrument in the facility, and as you can see, my users are quite happy, and the reason for that is because it's giving them practically everything that they wanted. It's very easy to use. It's very powerful. We have a lot of different colors to use, and especially is quite compact. So we can expand the capabilities in the lab for the instrumentation. But always we have a wish list for other things, and my wish list was on the sorter. So Beckman came back to me and said, Alfonso, what do you have as your wish list? Imagine that you are in Christmas, and what would you like to have in your sorter? So I put my list. As you can see, fast, sorted, reliable. We don't want breakdowns, cheap, flexible, sensitive. It should fit in my lab. Easy to use instrument and software, especially for the users, compatible with my analyzers. So one person that is using two software doesn't have to learn a completely different software. Easy to set up and track, reporting QC, auto compensation, auto alignment, auto drop delay, auto stream adjustment, auto cleaning, biohazards, remote access to engineers, as many things as possible. As you can see, the list is long. So they told me, what if you come to the toy shop? Why you don't come? Because I want to be on holidays. Okay, no, but we can arrange this, and you come and you can see and work with us to tell us your list of priorities in the toy shop. So I had the big opportunity to be one of the groups going, small group, reduced group, going to Suzhou, where the manufacturing of the CytoFLEX is. And we were together with Alexis Gonzalez, David Gabbard, and Peter O'Toole. So we went there to tell them things that as a core manager, we would like to see in this new prototype of the cell sorter. We were a couple of days with this fantastic team. We share ideas, we share information, we have discussions, and the prototype that you can see here later on became a reality. And this is more or less the size that occupies compared to the CytoFLEX S and the CytoFLEX LX. So it's a new member on the family. And I was quite happy to see that small space because, as James said, it's really good for my education programs because I really like to do trainings, and it is quite hard to move a cell sorter. But now, as you can see, I could easily have analyzers and sorter in the same room with no problem. They are portable devices that can be easily set up in a few hours. For the training course and for education, it's fantastic. So this is the situation now in the lab. So we have the sorter in the biosafety cabinet, as you can see, compared to the CytoFLEX S that we have here, how small the sorter is. As I said, there are lots of different things that we like to have, and one of them is that the instrument can be easily set up by the user. So there is one nozzle, there is the 100 micron nozzle. So when you press Start Up, it will ask you first to introduce the nozzle. So you can see on this video, click, done. So the only thing you have to do, as you can see, it's very big. You just plug it in, you hear the clack, and it's fit. Then they have to do QC. The user has to be a QC. It takes like less than 5 minutes, and it's the same quality control beads as the ones that we use for the CytoFLEX. And you can just basically reuse them for all the instruments that we have on the platform. It's looking at different things, that you have the right configuration, that you're looking at basically the performance of the instrument, laser power, the time delays, gains, coefficient of variation. And once this is finished, it's asking you, do you want me to calibrate as well for the sorting? So create these droplets and complete the side stream parameter setting. So in this case, it will be around 6 minutes. So I want to show you basically how this side stream is working. So you can see, we just say, yes, continue with the QC. It's looking at nozzle, frequency, amplitude, charging on the plates, and started to look which one is the right angle to select my angle, my cells. It's looking at the defanning and looking at the drop delay. This is done by sensors that they're localizing the system, and it's not using any kind of beads. So as I said, all the users can easily do all of this. You just click and it's done automatically. The advantage is that you can see you can manually calibrate the instrument if you wish. So it's done in such a way that for a user, a regular user, you do and automate everything. But if you're like a core manager or you are an expert in cytometry, cell sorting, and you want to manipulate things, you have also that option. The process to have the instrument starting up, doing all the cleaning, processing, sorting, calibration, quality control is less than 30 minutes. And in terms of the platforms that we can sort, you have on one side the 5 ml tubes, you have the 15 ml tubes, or different wells where you can collect the samples. Another advantage here, as you can see here, there's kind of a line or an angle. I'll come back to that one later on, which is the possibilities when we are sorting these 96 wells. So keep in mind these two possibilities here for collection of my sample. And then in terms of purity modes that we also have purity, enrichment. You can combine these, you can collect the aborts. And one of the beauties here is once we are trying to collect the sample, what we don't want is that when the tube is full, continue collecting your sample. Okay, so if we say here, which one is the tube size, so 5 mLs, or we can say it's a 15 ml tube, and we say which one is the initial volume, it will create here this gray bar telling you how much space you have on the tube left for collection of your sample, in this case indicated with the green color. So once it's reaching the maximum capacity, it will stop that collection. But it's also calculating how many of your cells you can collect to fill up that tube. So if you, instead of 1 million here, you say, I want to collect 10 million, it will tell you with this enrichment capability, 10 million in 5 ml, when you added 0.3 milliliters, it's impossible. So you will not make it. You need a bigger tube, otherwise we will not do the sorting. Okay, so it's telling you in advance what you can collect into that tube, or the maximum capability to collect cells into that tube. As I said, one of the things that we want is that the platform, the software itself, was very similar to the one that we have on the analyzer. Okay, so in this case, we are looking at a very similar software. As you can see here, this is SRT configuration for my analysis. And what we want to do is just one extra thing, sorter. So when we have this sorting, we can analyze my samples, and we have here the different tubes where we are collecting and sorting samples. Okay. One of the biggest advantage here when we are comparing my samples is, if you think I want to compare sample 1 and sample 2 and create now these gates, I have to go back and forth to modify those gates. One of the things we can do is exactly what we can do on the CytoFLEX analyzer, which is going to this second tab here. So you can see we have one tab that is for analysis, and we can analyze or reanalyze data on the second tab. In this second tab, we can copy and paste exactly the same graphs, but we can compare two samples at the same time. So this is wild type versus F8 sample. This is the one that we want to sort. We can build up this gate here, okay, based on my wild type. Or the other thing we can do is create an overlay of my wild type versus F8. We can afterwards develop our gate. We can copy that gate, and if we go back to the original one, the acquisition sorting platform, we can paste that gate. So this one is the pasted gate from my analysis gate. This is really advantage, because then we can use this when we are working with samples a bit more complex, where the limits are not easy to define, or when we are trying to do FMOs, for instance. Here we have one sample, CE9 label, in this case CFA, and now where am I double positive? You can create the polygon here, copy in this overlay, and transfer to your sorting platform, okay? So this is a big advantage to define the populations of your interest for sorting. Now, in terms of applications, what do you want to sort? So remember, if we can analyze anything, we can sort anything that we could see, right? So applications, open your mind. Transfection, cytotoxicity, cell proliferation, cell activation, calcium, microbiology, phagocytic assays, whatever you think that you want to sort, we can sort, okay? Even we can go for the nano sorting, and we will go into those details in a few minutes, okay? Cell types, any kind of cell types, any from mammalian cells going to apoptotic bodies, or the area of EVs, coming from cells from tissue or sperm, phytoplankton, or nanoparticles. So what we will do is trying to cover all this range with different applications that the different users that we have in the facility are bringing regularly to the platform. So let's start from viability. So in this case, this is from one of the users, Dr. Darrell Andrews, and she's working with viability. So they are trying to see the cytotoxic effect of different drugs. So in this case, you can clearly see we start forward side and scatter, then we added propionate iodide. So if we can clear up, in this case, our junk, debris versus my cells. When they are cells, we can see that we want to clear those ones. And then she wanted to do an assay. In this case, she didn't want to do sorting. She wanted to do just basically analysis of apoptosis. And instead of using the annexin 5, and we can see here the typical profile of annexin 5 versus propionate iodide displayed by ThermoFisher, she told me she wanted to use a new product that BioLegend has, which is ApoTracker Green. So when we look at ApoTracker Green, these are my alive cells, okay? And then these ones are the ones that they are expressing the phosphatidylserine externalized, and then the DNA is labeled with, in this case, helix NP, okay? So very similar profile to annexin 5, very easy assay, and when we analyze the sample ApoTracker with propionate iodide, we can see a very similar profile to annexin 5. But when we look at the live cells, and I was looking at the different populations, it didn't make sense. And I told her, look, for me, I'm looking at your kinetics, we are talking in a different things. This is not behaving the same way as they show here, and it's not behaving in the same way than the typical annexin 5 profile. So what we can see is that we have a live cells here. The early apoptotics are moving in one direction and then elevating. So it's taking, yes, apotracker, but then later on started to get more and more propionidolite. Here you have a bit more advanced apoptosis. Here is even more advanced, and now we are starting the DNA fragmentation. So this area here, they still have annexin 5 binding with apotracker, but they are dropping down in the content of propionidolite. Of course, this is my interpretation. Because it goes against this one, we need to verify it. So what we have done is looking at those different groups and sort them on the SRT. So here we can see how my fragments after sorting look like. And you see it's exactly in the same place than the analysis. When we look at the advanced apoptosis, you can see how these events are falling into my forward and cytoscatter. When we look at the apoptotic, we can see it's moving clearly in a higher forward scatter. And when we look at the live cells, it's clearly where we have the live cells. So the path that we see in this apotracker green doesn't correspond exactly in the same direction that we have on the annexin 5. Even if they are going through the same targets in the cells, the behavior of the dyes is not exactly the same than annexin 5. The other user that I want to point out is Dr. James Reynolds. So James is working with a completely different environment. In this case, he's working with transfected mice. They are transfected with TD tomato. And what they are doing is a contusion in the spinal cord. And afterwards, they are analyzing the damage on the sham, just on the day that they have done this process, 3 days after injury and 7 days after injury. And then they want to evaluate those cells by genomics. So in this case, yes, sorting is a need. So when we look at the tissue, we can see forward scatter and side scatter. You can see it's quite difficult to see which one is my target group of cells, the microglial cells. And what we know is that they are transfected. So they have the TD tomato. So it looks like these ones could be, but do we have different levels of these cell type, this TD tomato expression? So what we used was DRAG5 to identify those events that they have nuclei. So we can clearly see now here the nucleated cells with DRAG5 and combine nucleated cells, DRAG5, with TD tomato expression. So we can see these ones, they have DRAG5, but they do not express so much TD tomato. While these ones, they are my nucleated cells with the expression of TD tomato. Here we have other events, but they do not have the expression of DRAG5, so they are not nucleated. Okay, so these ones are definitely the ones that we want to collect and do sorting. DRAG5 positive, TD tomato expression. We make sure that we are working with singlets, and afterwards we sort them. Fantastic, easy. Now, beauty. As I said, we can do the overlays to verify my analysis, or we can verify or use the same software for reanalysis. So this is on day 0 when they do the laminectomy. Second, on 3 days afterwards, we can see clearly how these events have increased on their side scatter. And look like we have 2 different levels of expression. And finally, on day 7, what we can see is the side scatter remains higher, but the expression of the TD tomato is going even higher. So in this case, what we are trying to do is even further differentiate these kind of microglial cells, and of course, go into RNA analysis afterwards. Microbiology, let's go smaller now. So microbiology is an area that when we think on the amount of applications that we can do by flow cytometry is massive, but it's quite interesting because there are not many users in our facility in the microbiology area. So I hope this is starting to convince more people to use flow cytometry and cell sorting to work in that they are working in microbiology. So The only thing we need to do when we're working with bacteria or with yeast is just changing our mind. Flow cytometry is usually designed by working with mammalian cells, big events. So the default settings usually in forward side scatter are decided to work in mammalian cells. And even the threshold is decided to cut events that are smaller than different sizes. When we are working with yeast, these are still big. So we just modify slightly my side scatter and forward scatter. They will be on the range, but we can still work with the threshold on the forward scatter. But if we are working with bacteria, even increasing my values on forward and side scatter, you can see they do not reach the values that we have here on my side scatter on yeast or AP beads. And the other thing that you can find out is that when we're working with bacteria, usually what we will use is a threshold on side scatter, okay? So when you're working in this area, we have to play now in a different game, working with side scatter, or in this case, we are using a threshold or a triggering based on fluorescence. What we can do is similar to what I showed you before with the tomato. We can tag our events and gate on them or chop them. So in this case, we can use Cyto 9, for instance, or we can use this triggering based on the fluorescence. So if they don't have the fluorescence, we will not see it, okay? Be careful, because if we are talking about analysis, we don't see it, that's fantastic data that we don't process. But if we don't see it on the sorter, it's something that the instrument will ignore, and therefore we will not remove from our sorted population, okay? So be careful with this, because what we don't see, we don't sort out, okay? So this is one example with my microbiology, where we analyze these different populations, looking at protein iodide and the receptor of our interest, for instance. And what we have used is the CytoFLEX platform, and we have run this like in a 96 well plate. Okay, and we can see different profiles here showing the characteristics of the expression of these receptors, for instance, versus their viability. Okay, and then we can start to look if we have analyzed, but also the different levels of expression, alive death versus receptor positive, or we can look at the receptor expression only on those events that they're happy and alive. Okay, and then we can select those ones of my interest for further analysis. And in this case, we have this group of bacteria. We can see that we have some red spots. Those red spots are proteins that crystallize and produce that red fluorescence. So in this case, we can see our population of interest for one side scatter. These are my cells, how they look like, and the expression of this protein in the red fluorescence. When we look at these other ones, when they change the media, what we can see is two things. First, we have more red spots, which means we have more crystals. When we are looking at those crystals in my cells, you can see that the side scatter has been clearly modified. More side scatter will produce, but also because it's more red, we can see here the big increase on the red fluorescence. So ideally, what they wanted to get is the brightest signal that we can get from these ones. So sort those ones because they are the cells of their interest, the higher producers of these proteins. So let's sort them. As you can imagine, proteins on one side scatter on the other. My controls on one side and my controls on one side treated on the other. This is the population to be sorted. Now, things to consider. Cyto 9, first to remove them from the background. This is background, we don't want to take it. Looking at viability, in this case we use drag 7. Okay, so we haven't added yet. Fluorescence intensity versus viability. And finally, the same fluorescence intensity, but as a histogram. Okay, now we will look at how my population, when they have this media, looks like. So higher side scatter, we will have more fluorescence intensity, and we can clearly see those two levels of expression of this protein. Okay, but you can see as well the increase here on the side scatter, even with the Cyto 9 positive cells. But how many of them are alive? So when we put the drag 7, we can see that we have a lot of these events dying and they are DRAG7 positive. If we look at the same thing in my target ones, we can see a lot of DRAG7 positive cells that they are double positive as well here. When we look at the histogram, this is how it looks like without the DRAG7. If we look only at those events that they're alive and happy, if we started to gate them, we can see the profile change completely. Okay, so these proteins tend to aggregate looking like they were positive, but also we have a good amount of dead cells that they are coming with that protein as well, expressing the same level than this one. So all of these ones are dead. Only these ones are the ones that we are interested in collecting and sorting. So viability, when you are sorting, is one of the key things. Make sure that you are sorting alive cells. Otherwise, it's difficult that they grow afterwards. Of course, in the analysis, it's the same thing, same situation. So we have this level of expression of the proteins, and what we want to do is collect those ones which are the target population. So now, in terms of how we sort and how cool is or how easy is to sort bacteria, because there's contamination. This is something that we have done with a collaborator that we have in Poland, in Provi. In this case, they got the sorter. We have, as you can see, they have good fun, and Michał Mał here was preparing the samples just there. As you can see, sterility and the environment is quite low. Okay, what this group wanted to work with is, as you can see in this super formula, is probiotics. They're preparing probiotics, so they're very interested in the microbiology. And what we wanted to do is creating clones. So we selected singlets. If we want to do singlets, if we want to do cloning, we want just one bacteria, okay? And one of the things that we started to test first is this double population that we have here. They're not known. These are dead cells, no problem. These are alive cells, fantastic. But this group here in microbiology is recognized as countable but not culturable. So you know that the bacteria are there, but when you put them in the same media that these ones, they do not grow, okay? So can we use different medias, and if we change different medias, will they grow? So in this case, what we wanted to do is single cell cloning, but we want to collect 200 bacteria into each one of those positions. But the bacteria, usually we have agar plates to grow them, right? So what we will do is put this into an agar plate. So you can see an agar plate, we just have the holder, and this helps us as well to localize which ones are the positions of the groups that we are sorting. After sorting, you can see this is after sorting and after a few days of growth, how the alive ones are alive. And in this media, these not culturable but countable cells are not growing here. But the other thing that you can see is that those ones are not growing, but nothing else is growing in this agar plate. Only those ones that we place in, okay? We can do similar stuff. We can grow or select different populations that we don't know who are they. And afterwards, once we have the colonies well formed, we just move them into PCR and we can provide them the name. We can identify and come back. And now we know population 1 is this group of bacteria, population 2 is this other group of bacteria. And as I said, it's single cell, but I show you a presentation where we were sorting 200 cells. So it doesn't look like single cells. So let's go for a single cell sorting, and in this case by Theodora from System Biology Ireland. So singlets, scatter, second singlets, making sure that we don't have anything. And then she has been doing some transfection. In this case, it's M-cherry transfection. What we'll do is trying to see the transfection and collect those cells that they're positive transfected with M-cherry. The name of the experiment, it says everything. I hope it works, because the transfection efficiency was extremely poor. Being generous was 1%, okay? So very difficult sorting, okay? So she wanted to collect one single cell and clone them. Okay, so this is the target. Collect one single cell transfected into my 96 well plate and grow them afterwards. When you are thinking on single cell sorting, and this is trying to go back to what we said at the beginning on the design of the instrument, what we have is that usually the mainstream go directly into the waste. And then what we are collecting is those drops that we charge, and we put them into the 96 well plate, into the well that we want, okay? The problem is that sometimes those wells are empty. Even if we drop one cell, doesn't look like we have collected anything. And this is what we are talking about. They just bounce out, or they go because of they are collected because of the electrostatic charge of the plastic that are surrounding my well, okay? But we don't see them. So we have one option, and some of the companies are doing this, is basically slightly change the angle of the collection of the 96 well. But in this case, Beckman Coulter decided to do, or the team of Beckman Coulter decided to do, is basically put a new characteristic, which is called stream mode switch. What is a stream mode switch? So you click on this one, it tells you to remove the waste container. If you remember when I show you the waste container has two different positions, one straight down, one arrow straight down, and the other one in an angle. So when you're switching this, you are moving the waste to the left-hand side, and all the drops will be charged except the one that you are interested in collecting. That one is not charged and will be collected into your well. So that way, you're basically sorting in a straight line. No charging, so maximum effectivity. Of course, we are collecting the information as an index sorting because it's very useful afterwards to follow up what or further characteristics of your cells, or if you wanted to do a similar sorting than the day before, you know which ones are the best positions for collecting your cells. So we collected the index sorting. These are my populations of interest, imagine, and we just asked the software to show me in as rare events where are the cells of my interest, where they are coming from, okay? These are the cells that have been collected, where are they coming from? So Theodora, after a couple of weeks, came back and she said, I want to do a second sorting, and I want to verify as well what's the effective transfection that we have, okay? So this, for instance, was her Jurkat wild type cells, and now the M cherry that was coming from G9. So if we look here, G9 was collected, fantastic. Which one was the position of G9 versus the rest of the sample? So we look and we can identify G9 was very high efficiently transfected when we collected, okay? Really nice application. And as I said, one of the things I like about the Accuray and what I like in especially on the CytoFLEX is this large dynamic range, because it allows me to work with different cell lines, and the user doesn't have to move too many things. They don't need to manipulate too much the position of the voltage, in this case, gains of forward and side scatter. So to be able to know what's the size range I have in this dynamic range, what I've done is looking at different quality control bits that we have in the lab to see what are the range of populations that we can fit into easily into the SRT, CytoFLEX SRT, without changing my forward and side scatter. So in this case, I haven't changed forward and side scatter. I set it up, and then I started to record flow checks, 10 microns. Now we have flow sets, 3.6 micron. I analyze the CSNTs, 2 and 3 micrometers. Okay. Now, VersaComs, 3 and 3.4. UltraComs, 5 micrometers. It's quite interesting because UltraComs are 5 micrometers, okay, larger in forward scatter, but it's the same side scatter than the smaller CSNTs, and also smaller in side scatter than VersaComs and flow checks, scattering. Biocoms, these are coming from Slingshot. These are hydrogels, artificial cells, that they are 7 microns. So you can see forward scatter is smaller than the forward scatter of the UltraComs, okay? Then we have the Malvern, the yeast, which are silica beads, 0.4, 0.2, and 0.1. All these populations without changing these gains. These are the same gains. I just modify slightly the threshold to look more into the details of each one of those populations, okay? And finally, I ran the rainbow from Celarcus. You can see the different populations from very small particles to 10 micron particles, all in the same dynamic range. Why I'm interested in this? Because we have to look at different biological questions. And one of the ones is coming more and more popular is, Alfonso, I want to analyze and I want to sort EVs, extracellular vesicles. So as everyone in the field, we have the experience in some applications, but when they are talking about EVs, I feel like a baby, to be honest. So extracellular vesicles, for those ones that they haven't heard too much about EVs, they're usually originally considered as cellular debris, but it has found out that they have a good role in cell signaling, especially for molecular communication. These EVs are released by different cell types, from bacteria and yeast to lymphocytes. Okay, they reflect antigenic content from the cells that they are coming from. But usually, as I said, they were considered cellular debris, and this is because basically they contain things that the cells do not want. Misfolding proteins, cytotoxic agents, or metabolic waste. But they can also transport mRNA, miRNA, and proteins between the cells, and this will work as a good way of communicating cells and different parts of the body. Usually, we didn't pay attention to the CVs because it was considered as garbage, the junk that we were usually gating out. But because of this effect of molecular communication, and because it's involved in different, or has been seen that it's involved in different diseases, people is paying more attention. And usually, when I'm talking about EVs, I usually classify this as garbage, because it's basically garbage. But if we look into the detail, into the garbage, we can find information, very useful information. For instance, we can know what the neighbors are eating, what's the diet, the regular diet, if they're drinking Coke, or Pepsi, or if they prefer to eat pizza or handmade food. You can get very useful information by looking at that garbage. And that's what we are doing at the moment, and different groups are working is on the EV, looking at the garbage to identify biomarkers for different diseases. As I said during the description, it's a very heterogeneous population of events, and usually it's really important to analyze those events because they have implications. And that's why we are going into what is called the nanoflow cytometry. We need to identify my flow cytometry. We need to perform changes on my flow cytometry and the dynamics of my flow cytometer to understand what we can do and things to consider to do it correctly. Why? Because as you can see in this case, conventional flow cytometer has a limitation on the range of detection. Nowadays, we can see a series of different flow cytometers that they are dedicated flow cytometers, that they are able to go much lower in the number of events or the size of the events that we can detect and clearly separate from the other events and look at the different subpopulations. Of course, this one has a limitation as well. As you can see, other technologies are able to get a better distinction between the different subpopulations of EVs than flow cytometry. Okay. So when we're working on the nano flow cytometry for small particles, one of the first things we need to be careful with, and we haven't taken this in consideration until a few years ago, is what is called the swarming effect and the idea of removing aggregates. What is the swarming effect, and why we have to be careful with the swarming effect? The swarming effect is basically one small event could look like a massive event. And the reason for this is that, like in this case, this cat looked like a panther, or in this case, this amount of hundreds of different birds look like one big bird. Okay, so when we are analyzing by flow cytometry, we are looking at these as one single event, when in reality, there are thousands of them. Okay, so to avoid this swarming effect, what we need to do is dilute my sample population to analyze each one of them one by one. Of course, there could be still aggregation in here, so we need to find out the best way to remove those aggregates. How we will distinguish if we are working with aggregated events or single events? By looking at things like the median fluorescence intensity. We do a series of dilutions and we see how much this change. As you can see, the populations look completely different when we dilute them 1 in 5, or we dilute it in 1 in 20. Forward scatter, side scatters here, violet side scatter and blue side scatter, have dramatically dropped down on the values of the median fluorescence intensity, becoming more compact, and it's because we have now an analysis of 1 event. When we're looking at different samples, like in this case, sample of plasma from patient on day 3 and patient in day 1, and we do different dilutions, we can see the profiles clearly change when we do these dilutions between 1 in 50 and undiluted, or 1 in 50 and undiluted. Okay? So it's really important to look at those ones, and usually on the CytoFLEX analyzer, we have this possibility to look at the number of awards. So the lower the number of awards, closer we are to the reality of working with one single event instead of the swarming effect. In terms of removing the aggregates, usually my recommendation is, instead of using violet side scatter height, violet side scatter area, to work with violet side scatter height, violet side scatter width. The gate is easier to create in this plot. And when we look compared to this one, how singlets created by this violet side scatter width, violet side scatter height are, you can see very sharp line that it's impossible to create with this gating here with height and area. Okay? And clearly here you can see my aggregated particles. Okay? One of the things just before I move on is when we are working with very small particles, avoid vortexing. The reason for that is because you're creating air bubbles that they scatter light, so it will be looking like this instead of this. Okay, so be careful, do not vortex, just pipette up and down and gently. Okay, so as you can see, I was talking about violet side scatter. So first of all, why I haven't used forward scatter when we're looking at very small particles? And this is because usually on the forward scatter, what we have is an obscuration bar that blocks the direct light into my forward angle detector. So we are collecting the forward scatter. Okay. Now, when we are looking at side scatter, there is no such blocking bar. We collect the light directly reflected by the particle. And if we are thinking, usually we're working with blue side scatter, blue forward scatter. So the scatter from the blue laser. Now in the nano world, what is happening? The event is much smaller. And we have this blocking bar in the middle that doesn't allow us to correctly characterize how much scatter in the forward angle this particle is producing. Meanwhile, now what we will do is looking at in this paper the comparative of different instruments, okay, that they are trying to characterize 0.5 and 0.9 particles. You can see that one of them is not even able to differentiate 0.5 and 0.9 particles. If we look at side scatter, there's nothing blocking the light. And when we look at the similar instruments, so it's the same instrument now, but instead of using forward scatter, using side scatter, we can see that side scatter is not able just to differentiate 0.5 and 0.9. 0.1, 0.35 and 9, 1, 3, 5 and 9, 1, 3, 5 and 9. So even this one that is not able with forward scatter to differentiate 5 and 9, with the side scatter is able to identify easily these 4 different subpopulations. Okay? And why is this? Because usually what we need to look at this is not size and it's not density. Forward and side scatter, they're always related to my refraction index. Things like in cells, like viability or non-viability, will affect my refraction index. If we fix or do not fix the sample, or if we have different drug treatments, this will affect my side scatter and my forward scatter. Okay, so anything that could affect my refraction index will affect the values of my forward and side scatter, as I showed you before when we were looking at the different particles and how many of them we can differentiate. Okay, so Edwin Valdeport was working on this, and he proved this with the Mie theory, which is basically showing how much light we are scattering and the real diameter in this case of polystyrene beads. But if we change the polystyrene beads for any other beads with different refraction index, like the silica, the relationships between the scattering light and the size is different. And if we use vesicles that they have even smaller refraction index, we can see that the scattering intensity is also dropping down. So when we are talking about 200 micrometer particle of polystyrene, doesn't mean that it's equal to 200 micrometer vesicle size. We are talking that we have a much larger diameter in the vesicle for the same scatter. Okay. The other thing we can use for analysis of the EVs is using the fluorescence. Okay, we can label our EVs and analyze. And usually what we will do is using different dyes to further characterize the different subpopulations based on the cells that they are releasing these particles. So using antibodies tagged with a fluorescence. Okay. So, okay, this is cool information. Can you sort them? Can you analyze them? If I had to do it, what I should do? So first thing, remember, you have to do dilutions, okay? Second thing, as you can see on this technical note, this application note, we are trying to set up the CytoFLEX for EVs. What we'll have to do, looking at beads, calibration beads, and set up my violet side scatter. Okay, violet side scatter is more sensitive than the blue side scatter. Okay, try to thresholding, in this case using the violet side scatter, and making sure that we have the correct values for my blue side scatter and violet side scatter. Okay. When we look at this one, this is a different one, we can see that we can nicely separate different subpopulations of the EVs, as we can see here, 192, 500, or 700 nanometer particles by using the violet side scatter. This is a paper from Vasilis and John Tiggs. Okay, so what is changing the violet side scatter? Violet side scatter is something that you can do in practically every flow cytometer that has a violet laser, which is basically using this filter that we have here, the 405 filter. It's the same wavelength that the one we're using to excite my different dyes. So what we are doing in the CytoFLEX platform, analyzers and sorter, is basically swap the 405 filter to the position of the 450. If we need to use the 450, rather than placing it where we have the 405, we move it to the next filter. And if we need the 525, we move it to the violet 610, and we move this filter backwards. Okay, but the first position here has to be the 405. That one will give us our side scatter coming from the violet laser. Okay, so we can get the scattering and any fluorescence in the other channels. This is something that, for instance, Jordi Petriz has done a few years ago with the Attune NXT to differentiate the erythrocytes from platelets and leukocytes in the protocol that he established as no wash, no lyse. So this one is basically separating with the blue side scatter and the violet side scatter the population of erythrocytes from platelets and leukocytes, because they scatter completely differently, in this case, the erythrocytes from the other two populations. Okay? Right, so what about using, because my instrument has blue, violet, and red laser, can I use the same thing, same concept by using the red side scatter? So here's my red side scatter. I change my red side scatter for any position on the red detections block. And now I have an instrument that I have my SRT configured with violet side scatter, blue side scatter, and red side scatter. When we analyze the same samples that he's doing, no wash, no lies, into my CytoFLEX, I can see my blue side scatter, violet side scatter, blue versus red, violet versus red. And you can see the population of the erythrocytes is clearly different in two of these combinations. Okay, with the blue and red follows a diagonal. Okay, if we tag now the leukocytes with DRAG5 nucleated marker, what we can see is where the leukocytes are compared to the erythrocytes. Okay, now if we pay attention to the platelets, what we can see is that this combination of the different scatters is allowing me to resolve different populations of the platelets that we couldn't see before if we were using just forward side scatter or even with violet and blue side scatter. So as you can see here, we have the combination of the different dyes, of the different scatters, and you can see how different subpopulations are pulling out when we have the different proportions of each one of them in consideration. What if we try the same thing now with in the EV world? So extracellular vesicles, we said we can use different particles to calibrate the instrument, like in this case we have the megamix. Forward scatter, as we described, is not sensitive enough to differentiate these different subpopulations that we can see with the blue side scatter. And when we place red side scatter versus blue side scatter, we can see practically a good diagonal in the different subpopulations. Okay, when we run the EVs, we can see as well this diagonal with the red side scatter. Now, let's go to other beads, not the megamix. What if we look at the NIST, the silica beads of 300, 150, 180 nanometers? You can see here the blue side scatter differentiate two populations more than forward scatter. If we look at violet, we can differentiate from all of them, from the 80 to the 300 with no problems. And when we look at the red, oh, this looks the same than the blue, but it's slightly more sensitive than the blue. When we look at the Rosetta, it's interesting because with the Rosetta beads from ExCytometry, we can see blue, different populations, violet, different subpopulations, and red, different subpopulations. The interesting thing here is that red side scatter has more definition in the low end compared to the violet. You can easily resolve the red and the green. Not in the violet side scatter, you can do it better using the red side scatter. And for the bigger events, you have better resolution on the violet side scatter compared to the red side scatter, but it's even better on the blue side scatter. Okay? So look at the patterns between the Rosetta beads, okay, in the three different combinations of these different scatters versus the megamix, very similar profile. But when we compare this with the silica beads, you can see the profiles are slightly different. What I'm saying is when we are combining these different scatters, we can nicely separate and easier separate multiple subpopulations of these particles with more sensitivity. For instance, we have another set of beads like the apogees that when we look at the violet versus the red side scatter, or violet, blue, or red versus blue, we can see that there are populations that they're going in parallel. These set of beads, they have one characteristic, is that we have no label particles and we have labeled particles. And some of them are polystyrene beads and the other ones are silica beads. What if we select one of these diagonals? When we select one of those diagonals, they look like they are no label. If we look at the other subpopulation, the other diagonal, we can see that they are the labeled ones. Okay, so depending on the composition of my particles, looks like the refraction index could be different and the proportions of the scattering could be different. So what we have done is looking at red side scatter versus violet side scatter, again, doing the similar assay that I showed you before, but looking at the different subpopulations without changing the values of forward or any of the three side scatter. So we can see flow checks, calibrites, VersaComs. Look at this profile, very sharp drop down on the red scatter, very broad on the violet side scatter. When we look at the UltraComs, that they are larger than the VersaComs, they are coming smaller on the scatter. These ones, they change completely the relationship of violet versus red. When we look at the VioComs, the hydrogels 7, they have clearly more violet side scatter than the UltraCom beads. The Rosetta, we can see that they are following a completely different path. This is a mix of different beads. And again, we can see here that this set of beads, they have more red side scatter than violet side scatter. If we look at the silica beads, 0.4, share the same space than one of the Rosetta beads. And if we look at the 0.2, they're sharing a different space comparing to the Rosetta. Then we add as well red iron oxide from 0.3 to 0.8 are clearly scattering more red light than violet. When we look at the profiles, the same profiles on red versus blue, we can see it's very similar. But when we look at the violet versus the blue, we can see with that combination, we will miss a lot of those subpopulations. We will not be able to differentiate them. So I was not convinced that I was doing things right. That could be just peculiar of my instrument. So I double check things with John Tiggs in Beth Israel in Boston. And say, look, John, I'm looking at this. You have similar instrument. You have another CytoFLEX. Could you double check if what I'm seeing is correct? So these are my apogee compared in my analyzing my instrument. And these are the apogees analyzed by John Tiggs in Boston. As you can see, they have the same one. So, okay, now compare the apogees underneath and overlap them. As you can see, every one of these set of beads, they go everywhere in the different scatters. So what we did is I asked now this student I have, is an Erasmus student I have, Irene Castrosin. I told her, okay, you are working with me, and after 1.5 days training on the SRT, she hasn't done any sorting before. She hasn't set up the sorter before. She has done just one small training of 1.5 days. I said, look, turn on the SRT. Now configure the instrument, change the filters, and what we will do is analyzing apogees. You mix them up with Rosetta, and what we will identify is which ones are the populations of interest. We will collect these populations. Why? This one is or has the same red side scatter, but they have completely different violet side scatter values. And when we look at these ones and this one, they have two different red side scatters, but they are practically with the same value on the violet side scatter. Let's see if they fall back once we sort them into the same positions. So when we analyze each one of them after the sorting, you can see they are perfectly falling back into the same place. I really like this one, especially if we pay attention to this group here. This is what the composition of all of them, but now here you can see this group sorted is clearly different than this one or any of these two. So clearly we can select these subpopulations that are around 200, 300 nanometer size and differentiate them based on their scatter characteristics. Nothing more than scatter. If this is working with cells, so we will check with cells. So we move now into cells in culture. We look at forward and side. We label them with drag7 to look at the dead cells. And when we look at the scatter with the different combinations, violet, blue, diagonal, but when we look with the red combination, we can see with more detail a bit here, we can see clearly two different subpopulations. The one in blue with lower values on the scatter correspond to alive cells. The ones with higher scattering correspond to dead cells. Is this an artifact or not? So I ask again, Irene, to sort these two subpopulations and reanalyze the sample afterwards. So she set up the SRT, sort those two populations, and reanalyze afterwards. So as you can see, the dead cells after sorting, they remain as dead with the same characteristics as we had before. And now the alive cells, they remain with the same characteristics on the scatter alive. The beauty as well is that, as you can see after sorting, we can see that all my cells have survived the sorting process. That means that this SRT is very gentle with my cells. We try the same thing with these artificial cells from a slingshot, because what we have seen analyzing them is that they look like they have two values on the side scatter, like two subpopulations, especially clear on the red scatter, not so clear on the violet side scatter. When we combine them, it's interesting because we can easily identify 4 subpopulations. When we sort them and reanalyze afterwards, we can see that they came back to the same distribution. So it looks like these ones are not 4 different distinct populations. These are the same ones, and depending on how they pass through the laser beam, they scatter in different ways. And this is bringing us to look at how small can you sort on the SRT, and how good are you separating these particles small. So I had access to these 60 nanometer particles from the NIST, the National Institute of Standard and Technology. So in theory, they are very homogeneous 60 nanometer particles. And after calibrating the SRT, we look PBS, and afterwards, we analyze these 60 nanometer particles. By looking at this, we can see that it doesn't look so uniform. Even if we have done all the dilutions, with the maximum dilution we got exactly the same distribution, different populations. When we look at red and violet scatter, we can see 1, 2, 3, and 4 different subpopulations of gold nanoparticles. In theory, they are all at 60 nanometers. So I ask again the student to separate these populations. This is the PBS that we had, and now PBS plus my first population, second population, third population, and my fourth population. Clearly, this one here is a distinct population because it has different red side scatter, violet side scatter than this other one. We have tried the same thing with phytoplankton. We analyze violet side scatter versus red side scatter with phytoplankton. And as you can see, different species, different mix, has different combinations of scattering, which is absolutely normal when we think the high variety of morphologies, high variety of contents, composition of cell walls, if they have cell walls, morphologies, etc. So clearly, these groups here with scatter will help us to sort them and further analyze these characteristics. And this is without considering that they also have autofluorescence. So the combination of scattering plus autofluorescence will help us to further distinguish and further characterize these subpopulations of phytoplankton or even zooplankton. Same thing, same concept can be applied to the extracellular vesicles. If you remember, we were talking about the garbage. These compositions, these cargo and content, they have different sizes, but also the structure of the membrane could be different, and the content that they have could be completely different. So can we use this scatter in our favor to characterize my EV populations? Because once we see them, we can separate them, we can sort them, and analyze afterwards the proteins and mRNA, so even further characterize this. So let's see what we can see at the moment. So as I mentioned before, phytoplankton is here. When we mix them up with yeast, look at this one. Yeast are coming in completely different angle. And if I use monads, which are spores of this yeast, you can see that they're coming again into a different angle. Whatever you see, we can sort. So we can sort and further characterize. Same thing with the extracellular vesicles. In this case, what we have is 2 different samples from the same patient, day 1 and day 3, with markers of interest like annexin 5 and CD markers with double positive. And we can analyze how many of them are expressing, how many of them are co-expressing, and how are they looking like. So when we look at the red versus violet side scatter, and we overlap the samples on day 1 and 3, what we can see is if we gauge on the different subpopulations, excluding these ones that do look more an outlier, okay, that when we focus into this first group, they share on day 1 and 3, the CD marker expression of both of them are showing 2 different levels of expression, dim and bright, bright and dim. Now, if I move to this section here with high or higher red side scatter, that looks like on day 3 is gone, or practically no present, what will happen? Look, that population is highly present or high expression on this CD marker. Okay, it's quite interesting. Here is the same place. Most interesting is when we compare to this other population, population 4, that has less red side scatter. That population looks like, keep an eye here, or keep an eye here, looks like when we move to higher red side scatter, occupies the center position. So the expression of this marker is intermediate, and that makes this group of EVs slightly different and reflecting the light in a different proportions of red versus violet. Of course, if we go up in terms of scattering, cells are, EVs are entirely larger, so they have higher expression. And if we go even higher, higher expression of that marker. But when we go to this population here that has more red side scatter and has appeared on day 3, was not present on day 1, what we can see is that they do not express that marker. Even with the high presence here of these EVs on day 3, they do not show up any expression compared to this population that has equal red-violet scattering. These are platelet-derived EVs. As I said, what we can do is looking at PBS, no label, label with one marker, label with the other, and looking at the double positive. When we look at the double positive, we can see a very good amount of different expression patterns of both of them markers. What is nice is when we look at these expression markers and how much they express this as CD9 and CFSA, when we look at each one independently and we look at the scatter proportions of red and violet, we can see that they have a good variety of expressions on their scatter. So they are not uniform EVs. Can you sort EVs? Can you do it with ESRT? Yes, we can. And again, Irene, Erasmus student with very low, very low experience in cell sorting, set up the instrument, prepared these EV samples, and I asked her to sort those 2 populations, for instance. We have sorted all of them, but I will show you just 2 of them. And you can see the reanalysis of my population that we call large, and the expression on the scattering, and the one that we call weird, in this case, and how is their expression on the scatter. Clearly separated both of them, and you're thinking, what is this? We always have the PBS and the background noise, okay? So this is how there was the original sample, and then when we look at how these 2 populations look like after sorting, we can clearly see that they are perfectly distinguished, and we can sort them. And if we can sort them and isolate from the others, we can start to look at doing proteomics of genomics of individualized groups of EVs. Now, how they look like on the red side scatter, look how this weird population has a clear, distinct pattern on the side scatter. And if you look at this one that I'm pointing out here, it's like the gap that we are missing in this one. And when we look at the expression of this one, it was this group down here. So we have different populations that we can further characterize by the combination of side scatters, multiple side scatter, multiple markers, and once we analyze them, we can easily sort them with CytoFLEX SRT because of their sensitivity. So as you can see, there are loads of advantages using multiple scatter and flow cytometry for further characterization of EVs. We cannot just analyze them. Now with the CytoFLEX SRT, we can also separate them and doing cell sorting. As I said, and James mentioned, I really like to do workshops. I enjoy doing workshops and training and education everywhere. These are some of the examples. So we ran the summer school here at UCD, the winter and summer flow cytometry schools. We have run last year the first extracellular vesicle course here in Ireland, and we also do training courses in places like Poland, where now with the CytoFLEX SRT, we can combine not just training courses on the analysis, but also we can do training on cell sorting because, as you can see, it's a portable unit, very flexible for any kind of applications and very sensitive as well. With this, I want to acknowledge a lot of people that has helped me to perform a lot of things and learn a lot from them. So I don't know where to start because there are loads of different people that I have to acknowledge. Of course, I will start with the group from Beckman Coulter. As you can see, I just mentioned a few, but not all of them. So apologize if I don't put your name. The group, of course, of LabPlan, that they are our Beckman Coulter distributor here in Ireland, and people like John Tiggs, Jonita, Vanessa, all the group in Beth Israel that we are working side by side in different things. Peter O'Toole and Karen Hogg in the University of York, and of course the team in Sam Provi in Poland, Joanna and Kasia. And I cannot forget about all the users at the Flow Cytometry Core Technology at UCD Conway Institute, because without them, I will not have access to all these samples. They are very supportive. I propose them things, they provide samples, they test things, and they also allow me to share this information with all of you. And if you have any questions, any technical questions, even from the explanations I said, and I can put you in connection with all my users, I'll be happy to do it because that would create collaborations and further knowledge. With this, I just want to say thank you again to all of you for paying attention. Apologize if the talk was too long, but still happy to answer any of your questions that you have. Thank you, Beckman Coulter, for giving me this opportunity. And that's it. Thank you very much.
Dr. Blanco, thank you so much for that informative presentation. It was great. It was definitely detailed. And welcome back, James. So yes, it is now time for our live Q&A portion of our webinar. To our audience, if you have a question you'd like to ask and you have not done so, please do so now. Just click on that Ask a Question box, and it's located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, before we jump in, I just want to say, Dr. Blanco, I see you on camera, but it's not activating. Did you by any chance turn it off?
I'm doing my best.
From your I hear you. Let's see. Did you mute? You can always do a quick refresh in your studio. We're happy to wait. We've got some great questions already coming in, so we're going to dive in with those. Let's see. All right.
Have we lost off? There he is.
Can you hear me?
Yeah, I hear you. I don't hear Cassie anymore, but maybe.
Let's see. James, can you hear me okay?
Yes, now I hear you.
Yes. Yes, we can. No.
Am I the one frozen?
Uh-oh. You know what?
Cameras.
Maybe I'll take over for just a moment while Cassie's having troubles.
There we go.
Oh,
okay.
All right, there we go. I think I was the frozen one for a second here. Okay. All right.
Always happens at transition points, doesn't it?
You know, it sure does sometimes, and technology is great until it isn't, right? But okay.
Everything. We're used to that.
Well, we're just making sure you're all paying attention. So keeping you awake and on your toes. Dr. Blanco, let's dive into this question. What do you say? You ready?
Yes, I'm ready. Yeah.
Okay, so we do have quite a few questions that have already come in, and I'd like to start with this one. Do you have any concerns about not having other tip sizes and pressures?
That was a discussion point when we were in SUSO. When we were talking with the engineers, we always want to have different tips, especially in a core facility. We want the very small ones. We were thinking on having a 15 micron nozzle and all the way to 220, because we have people working with very small things, and then we have very big things, very large things. So the discussion was there, but we were discussing, and at the end we agreed that having one single nozzle that covers most of the applications. That's what we would be happy to have in a core facility to make a very easy to use instrument. If we started to change nozzle size, it means that we have to change the pressure, and this cannot be as operative as this one, because there are many things in the background run by the software that has to be readjusted, like the pressure has to be readjusted. So that will create a few more difficulties on trying to make a very easy to use instrument. For most of our applications, 80–85% of the applications that we're working on, there are transected cells, cells that they are quite fragile, cells in culture that we have trypsinize. So 100 nozzle is usually what we are using, even with our FACS area. Only when we are working with lymphocytes, for instance, we will go to 70 micron nozzle, or if we are working with bacteria, we'll go down to 70 plants. With 100, most of the samples that we have analyzed, they are fine with a 100 nozzle. Of course, if I were David Gabrith, who was there, he wanted very, very big ones. So we have to think on a different kind of instrument. And that's why we have both of them here. We have the SRT and we have a FACS area. Whatever we cannot do in one, we can do with the other. And again, think that with 100, it's good for most of the applications. And this is trying to find a niche in very small instruments in labs that they are not well used to have a very big core facility close by. So I think this is a good decision to have just one to simplify the process.
Thank you, Dr. Blanco. And I'm seeing several questions coming in regarding contamination concerns. How do you deal with contamination concerns running bacteria, yeast, and mammalian samples?
Okay, right. I'm also well known because I'm a cowboy, so I try everything with no concerns. So that's the reality. I've been here in Conway for the last 19 years and a half. We know that because we have celebrated now the 20th anniversary of the Conway Institute. So I was just at the beginning, and in these 19 years, we only had one contamination. We have worked with all these variety, as you can see. We only have one contamination on the sorting, and that was due to a sheet filter that was contaminated. So we replaced that one, and that's fine. But the other thing that we have, and in fact, it's funny because I got now 2 new nozzles here, so I still haven't put them in the lab. So the one of the things, as you can see, they're quite large. I will show one. It's quite large. So if we have users with this kind of concerns, they can buy their own nozzle and hold in, and then they just use their own nozzle, which could be one of the main sources of contamination. But there are lots of procedures and cleaning procedures to make sure that there's no contamination from the sorter side. And you have seen we are working with yeast, with bacteria, and many different kind of samples. And usually contamination is not one of our biggest concerns.
Very good. So one of our attendees says, I heard that area scaling is a parameter that should be looked into before gating your samples. What is area scaling useful for, and can that be verified with the CytoFLEX cytometers?
Yep. So area scaling, fantastic. Thank you for that question. I think that's coming from Angela Quach, Concordia University. So yeah, area scaling, if you think, and if you look back again on my presentation, I was working with area and height. And usually for mammalian cells, the recommendation is working with the area of the signal because they are very large. So when we're analyzing the sample, we're looking at electrical pulse created by the cell when it's passing through the laser beam. So we're generating a pulse. This is defined by the height of the signal, the width of the signal, and then the area of the signal. The reason for calculating the area is because we want to integrate the entire signal of the sample when it's passing through. This is with big cells. And of course, if you look at cell cycle, it's a bigger reason to work on area. Height, if you see, I'm working with height when I'm working with spheres, or I'm working with very small particles like bacteria or extracellular vesicles, because I find out it's far more precise working with this one because it's the measure of the signal when they pass through the laser beam. So it's just pure measurement. Meanwhile, area is calculated by the integral of the entire pulsing. So yes, the area scaling has to be adjusted because it's calculated from the height. And this needs to be calculated because if the height is too high, so if we saturate the signal, then the relationship of area height is not linear. We lose the linearity between the area and the height. If the signal is too low, we can lose as well resolution. So we need to increase a little bit more that multiplication factor. So we have to be working between the area and the height and trying to establish when we are in the linearity between the area and the height. The beauty of the CytoFLEX platform is that we have a very large dynamic range, and during this large dynamic range, we have a very close relationship between the area and height on the relationship, the linear relationship. To make sure that we are working and trying to avoid exactly this, the need for scaling, to rescale this area-height relationship, we need to make sure that we are working in the sensitive area where the area and the height are linear. So that's why we are requesting people to do the gain tracing on the instruments. There are gains that they are below this side, and there are gains that they are really high, and then this kind of relationship could be lost because we have a saturation of the height. Of course, it's quite variable as well between the samples, but usually a good range is working with gains between 100 and 1000. Below and above, I will be a bit more concerned. So again, what you need to do with CytoFLEX is doing a gain tracing to make sure that you have your system in the most sensitive area where you have the maximum sensitivity, and also you have a linearity between area and height.
Very good. Well, thank you for that. So, Dr. Blanco, we have a lot of questions coming in from people who want to get started. So you list a lot of controls for finding scatter sensitivity. Now, if a group is wanting to get started, are there one or two sets that they should start with?
Yeah. So flow cytometry and samples, controls, and controls. We need every kind of control that you can imagine, especially if we are going quite small. We need to start to increase the number of controls. We need controls for everything. So if you see there, when I was setting up the instrument, the recommendation is to use calibration beads where we know the sizes of this one. We use the violet side scatter. Usually we'll use a combination of violet side scatter with the fluorescence, because the fluorescence will be helping us to establish how low we could go, and then we'll use the side scatter. And that will establish a minimum point where from there down, it's quite difficult to distinguish my real signal from my background noise. Usually, this is something we'll use with apogee beads or megamids or the NIST. To make sure that we have that, we have to clean the instrument, make sure that the flow cell is clean, all the fluids are clean, the sheath filter is clean, our sheath is as clean as possible. So that's really important. Looking at the sheath afterwards, make sure that we have a really clean sheath, especially a PBS where we have to resuspend our samples. So getting a freshly made and filtered PBS is one of the key things. And when we are looking at antibodies, fluorochromes, dyes, when we're looking at cells, we always recommend titration. So titration control for EVs, extracellular vesicles, is recommended, but there's another control that we have to add, which is basically making sure that these antibodies are not getting together, that we don't have aggregates of these antibodies. Because when we have the aggregation of these antibodies, they will have similar scattering than by particles. So again, this is something we have to take in consideration. So we need to centrifuge the antibodies, remove all these aggregates by collecting just the supernatant, and run the supernatant in the middle of the PBS to know which one is the background coming from the PBS. So there are a lot of controls. I know timing is an issue, but this time is also really important. We need to make sure that the instrument is stable as possible. That's another background time that we need to check. And all these instruments, especially the pressurized systems, they tend to have a good amount of jump in these peristaltic, in these pressurized systems. And we'll need time to make sure that the system is as stable as possible to make sure that we get a good measurement. So again, time as a parameter is mandatory. So as you can see, there are loads of areas where we have to make sure that we control all the external things that could affect my readings.
Great advice. Well, in the interest of time, we're going to wrap with that final question. Dr. Blanco, I want to thank you for this excellent Q&A session. Sorry, James, we didn't have any questions where you could continue.
That's okay. That's okay.
I talk a lot. Sorry, James.
No. So make my life easy, Alfonso. I appreciate that. Thank you.
Welcome.
But I would like to give you both an opportunity to give any final comments to our audience.
James, I leave it to you.
Okay. Yeah. Thank you, everyone, for coming. I really appreciate it. Thank you, Alfonso, for all the great information. And thank you, Christina, for hosting. And I'll see you next time. If you catch me at a meeting somewhere, come and say hi.
Same for me. You can pick me up anytime, anywhere.
Well, very good. Dr. Blanco, thank you for joining us today, and you as well. James, it's been a pleasure to have you both on with us today. I do want to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I do want to thank our audience for joining us today and for their interesting questions. The questions that we did not have time for today, and additionally those submitted during the on-demand period, they will be addressed by our speakers via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will be sending you an email when it's available for replay. We encourage you to share that email with your friends and your colleagues who may have missed today's live event. Thank you again for joining us. Have a great day, everyone, and we'll see you soon. Bye-bye.
Thanks, guys. Bye-bye.
Bye-bye.